Welcome back. Hey, our next speaker is Snigdha Guha, and she is a graduate research assistant at the University of Nebraska in Lincoln. Snigdha grew up in New Delhi, was educated there, did her bachelor's and a master's degree there, and she's currently working on the role of dietary bioactive peptides in modulating metabolic disease. This includes looking at bean-derived bioactive peptides, efficacy to reduce vascular inflammation and related pathophysiology. So the title of Snigdha's talk is Efficacy of Dietary Gamma-Glutamyl Peptide in Modulating Vascular Inflammation. And her co-author is Kostov Mahumir, who's the assistant professor at Nebraska. So welcome very much, and we look forward to hearing your talk.
Thank you. Hello, everyone. Today I'm going to pitch for my research, which is titled Efficacy of Dietary Gamma-Glutamyl Peptide in Modulating Vascular Inflammation. So let me start my pitch by asking this one question. What is the #1 cause of death in the US? And the answer is cardiovascular diseases or CVDs. In fact, it is the #1 cause of death globally. And one fun fact here is that it has been found that if a person follow all of these 4, that is, doesn't smoke, controls their BMI, eat lots of fruits and vegetables, and exercise on a regular basis, they can actually reduce the risk of CVD by 85%. But sadly, only 3% of Americans have been reported to follow all these 4. And according to WHO, it has been reported that 1 in every 4 deaths in the US is because of cardiovascular disease. The picture here on the right is a summary which signifies the importance of inflammation in causing heart disease, because low-grade chronic inflammation is the main initiator of endothelial dysfunction or plaque formations in the blood vessel, which actually leads to disorders like atherosclerosis or hypertension, which are the major risk factors for cardiovascular disease. Now, the problem with the current pharmacological interventions for CVDs are that they have various side effects, as well as in severe cases, patients have to undergo vascular surgeries, which are very expensive. Therefore, in order to control such a growing epidemic, we need more of a preventative approach. And for that, my research proposes use of food-derived bioactive peptides. And one class of those peptides are gamma-glutamyl peptides. So these gamma-glutamyl peptides could be naturally found in foods such as beans, cheese, onion. And it has been seen that when gamma-glutamyl peptides interact with the calcium-sensing receptors or CASAR, which are on our tongues, they give rise to a kukumi flavor. And this same interaction with the CASAR has also shown to have anti-inflammatory effects, which was previously proven in gastrointestinal inflammation. And since calcium-sensing receptors are ubiquitously present in many mammalian tissues, including vasculature, therefore my research hypothesizes that gamma-glutamyl peptides can reduce vascular inflammation as well in CASAR-dependent pathways. So this is a summary of my research method and results. So for my research, I chose gamma-glutamyl valine or gamma-EV as my model gamma peptide. And I cultured human aortic endothelial cells, which are blood vessel cells from the aorta. And I first pre-treated them with gamma-EV for 2 hours at different doses, like 0.01, 0.11 millimolar. And then I induced inflammation using tumor necrosis factor alpha for another 6 hours. So when inflammation is induced, there is increase in expression of inflammatory biomarkers like selectin, cellular adhesion molecules, chemokine, and cytokines like IL-6, IL-8. But it was found that in the presence of 1 millimolar gamma EV dose, the expression of these biomarkers was significantly reduced, suggesting that gamma EV had anti-inflammatory role. Then we also found that this anti-inflammatory effect was calcium sensing receptor dependent, because when we used a CASAR specific antagonist, which was NPS 2143, which specifically blocks CASAR, the anti-inflammatory effect which we previously saw was abolished. We also looked at some key signaling molecules which might be involved in this mechanism, which included beta-arrestin, CASAR itself, then P65, which is a molecule of the NF-kappa B pathway, as well as Cre binding protein. The next part of my research focused on seeing whether this gamma peptide could actually cross the intestinal cells, because it is important to see if we consume them, they should be able to pass intact and reach the blood vessels. So for that, we grew intestinal epithelial cells CACO2 on permeable inserts and added gamma EV on the apical side, and then allowed them to transport for 2 hours, after which we looked their or checked their concentration in the basolateral layer. And we found that gamma EV did pass intact through the cells. We also looked at the mechanism of transport of gamma EV, and it was found to be both paracellular, which was between the cells through tight junctions, as well as through peptide-1, which is through a peptide transporter. Therefore, the relevance or the long-term goal of this research is, with such promising results, we could actually use gamma peptides to develop nutraceuticals or use it as a functional food ingredient, which would be effective in reducing vascular inflammation, and therefore might provide a novel strategy for prevention of cardiovascular diseases. Thank you so much for your attention, and I can take some questions now.
Well, thanks, Snigdha. That was a great presentation, and what a very interesting topic. Again, an example how food can have significant impacts on physiology in our lives. And, makes me want to eat more beans. So, but again, we'll have the judges ask you questions, and we'll go from there.
So this is Holiday. Nice job presenting, and I like the fact that you're using plant-based interventions. So that's awesome. I just had a question about your results. What did you find? Tell us more about kind of some of the conclusions that were made from the work that you did.
Okay, so we started with a single peptide, which is gamma glutamyl EV or gamma glutamyl valine, which was previously reported to show anti-inflammatory effect in gastrointestinal inflammation. So we wanted to see whether this peptide could actually also cause reduction in vascular inflammation. And so we did see that there was a dose-dependent effect for this peptide to reduce inflammation in endothelial cells, and it was effectively able to transport across the intestinal cells. So moving forward from here, since this is just one peptide, and as my research focuses on bean-derived peptides, second part of my research also involves with extracting these peptides naturally from beans and seeing their efficacy. So there would be like a mixture of gamma glutamyl peptides, which we have already extracted from beans. And eventually, and we have seen that they can reduce inflammation in vascular cells. So they might be used later on for as an ingredient for functional food, which something like diet, like supplements, which you can take, and then it would be helpful in prevention of these cardiovascular diseases.
Thank you. Out of curiosity, what types of beans?
We work with Great Northern beans. That's the most produced variety in Nebraska.
Okay, good to know. Thank you.
Yep.
Thanks a lot for your presentation. Very interesting. In the bean, these peptides, they exist as a peptides or as a part of a protein?
So yes, these peptides, they actually exist as peptides. So they are part of non-protein nitrogen sources. So you can actually extract them as it is. So when we extracted them, we did not digest them or anything. They were just present naturally as peptides.
Okay, and during the digestion, so let's say that you ingest these peptides, do you think that they would reach the gut?
Actually, we have not checked that yet, but in literature, it is suggested that because these peptides are gamma glutamyl peptides, and because of their unique gamma bond, which is unlike the usual alpha, which is like the alpha carbon, which forms the peptide bond, they form from the gamma carbon of the glutamic acid. So because of that unusual bond, they're actually resistant to gastric digestion. So they can survive the digestion which is happening in our gut and can actually cross the intestine and reach the blood vessel. So that way it is pretty unique as well to be used as an ingredient.
Thank you very much.
Okay, and a couple of questions from me. You mentioned earlier that they produce a kokumi flavor. Could you describe what that flavor is?
So yes, it's called the sixth flavor. So when you eat food like miso soup, if you know, they have that what do you call umami taste. Do you know that?
Yep.
So basically it was actually first discovered in miso soup. So these peptides were found to be the major constituents of the soup, and then there was research which identified these peptides and also linked them to their interaction with the calcium sensing receptors on our tongue, which was the main reason for these for the perception of this taste. And then later on, the studies showed their anti-inflammatory effects as well.
And following on from that, so you say cheese, beans, and onions are good sources. What is the best source? If you've got to name a source, what bean or cheese or?
For example, I have been working with Great Northern beans, but we have also looked at some other beans such as garbanzo beans, and I found that the gamma peptides were a little lesser than that. So I think in order to give a complete answer, we need to test all beans because their concentration would vary among beans. I have just worked with 2 beans, and I can say Great Northern beans really have a good amount of gamma glutamyl peptides, which is not so much in, for example, garbanzo beans. So, and yeah, but I haven't like looked at all the sources. So,
Great, thank you.
Okay, how much? What kind of concentration are we looking at? Like micrograms per gram?
Actually, in beans, it's nanograms. So it is, to give you a number, it's like 53 nanograms per gram. I know the concentration is very low. So few of the limitations is, one of the limitation is actually that. And then to overcome that, we might even look at some processing, like for example, fermentation of the beans has been shown to increase these gamma peptides in literature, which is another side of the project, which is going on currently in our lab, to see if by any processing methods we can actually increase the content of gamma peptides. But this is just like a fundamental step or like a starting point just to see if these peptides are biologically active or not. But then once we find that, there will be like other processing methods to actually increase their concentration, maybe breeding plants in a way to have these more. So yeah, but currently it's pretty low.
It's just curious. It's fascinating to me. I know I've seen some studies with fish dipeptides that have big effects on vascular responsiveness. So this question is from the audience. Do you think adding 1 millimolar of your peptide is high?
It is not high because we also looked at the toxicity on cells, and it was not toxic to the cells at all. And so we looked at a broad range, and we actually went up to 5 millimolar. And like after 5 millimolar, it was actually toxic to the cells, but 1 millimolar was not that high.
Thank you. And then a second question for you, Snigdha, was I think you addressed it a little bit. Can you scale up the production of gamma peptides with fermentation, like the example, making bread?
Yes. So that is some of the projects. So for example, we would try to do a bean paste or something with like more of a fermented product, which can be used. So maybe like bread, but so that would also require like product development side to it. But for now, maybe like fermentation to produce some bean paste or something would be is what I can see can be done, because fermentations using lactobacillus has been shown to increase gamma peptides. But yeah, definitely bread would be an option as well, I guess.
Okay. Excellent. And that's the extent of the questions from the audience.